Let's now talk about oxidative phosphorylation in the electron transport chain, or system, as it is now called ETS. First, what is the ETS? It is a series of enzymes. Look at this picture. I want you to notice several things. First, the yellow part represents the mitochondrial matrix. That is the middle of the mitochondrion, and it is filled with fluid similar to cytoplasm. Next, the mitochondrion is an organelle that has a double bilayer phospholipid membrane. These phospholipid membranes are shown here, as green rows with yellow edges to show the phospholipid heads. There is an inner mitochondrial membrane and an outer mitochondrial membrane. The pink or salmon color between the two membranes is the intermembrane space. The light blue region of the picture represents the cell's cytoplasm. Next, I want you to see that the enzymes involved in the electron transport system are embedded in the inner mitochondrial membrane. Next, notice the small inset picture. You will see the artist drew the mitochondrion, and it looks sort of like a giant clam, at least to me. Really, the artist is trying to show you that the inner mitochondrial membrane is highly convoluted, and this increases surface area so that many electron transport chain enzymes can be inserted. More enzymes means more ability to cash in NADH and FADH2 for ATP. So what happens during the electron transport system? Well, simply put, this is where we cash in our NADH and FADH2. There are varying reports on how many ATP each cashes in for. Some reports say 2, others say 3 for NADH. Some reports say 1, others say 2 for FADH2. Most modern biochemistry books are saying this: depending on the energy needs of the cell, each NADH can generate 2 to 3 ATP, and FADH2 can generate 1 to 2 ATP in the ETS. We will use the middle of the road. One NADH will generate 2.5 ATP, and one FADH2 will generate 1.5 ATP. Now, let's say we are ready to cash in one NADH. Well, NADH enters the electron transport chain at the beginning, and as it is passed down the chain like a hot potato, its hydrogen and electron are slowly extracted. Again, we don't want to end up like the Space Shuttle Challenger. The hydrogen is released into the intermembrane space, and the electron is ultimately given to oxygen. At the end of the chain, you should see a blue enzyme called ATP synthase. This clever enzyme capitalizes on the hydrogen gradient generated by the upstream ETS enzymes. There is a high concentration of hydrogen in the intermembrane space and a low concentration in the matrix. So, ATP synthase allows passive diffusion of hydrogen into the matrix, and as it does, the ATP synthase uses this released energy to push an inorganic phosphate group onto ADP to create ATP. The hydrogen that diffuses into the matrix happily joins with oxygen and reunites with its donated electron from earlier on to create water. I urge you to visit the website at the top of the screen. Just cut and paste it into your browser. There you will find a wonderful movie of the electron transport system. Do not memorize each ETS enzyme nor the cytochromes. Just understand the overall process as I described here. It is enough. Why does FADH2 only generate 1.5 ATP? If you look at this picture, you will realize that FADH2 does not enter the ETS at the beginning, rather further on in the electron transport chain. Therefore, there is less transfer of energy and less ATP synthesis as a result. Let's use this slide as an overview of all three metabolic pathways. Again, I want to reiterate that NAD and FADH are merely temporary storage molecules for hydrogen and its electron. In the electron transport chain, they cash in the hydrogen and electron, and ATP is made. There are varying reports as to how many ATP are made. It is safe to say that depending on the energy needs of the cell, that as few as 2 ATP to as many as 3 ATP can be made for every NADH. And for FADH2, the range is 1 to 2 ATP. Therefore, I will use the middle-of-the-road values of 2.5 ATP for every NADH and 1.5 for every FADH2. For you chemists out there, this stoichiometry may make you cringe, and I want you to know I share your pain. But let's just shoulder up and get through this. Let's start our overview with pyruvic acid entering the mitochondrion. There is a preparatory phase to convert pyruvic acid into acetyl-CoA before it can play on the merry-go-round of the TCA cycle. This preparatory phase generates 1 NADH for every pyruvic acid, and this means for one glucose molecule, this preparatory phase gives us 2 NADH. For every acetyl-CoA we make, this will generate 3 NADH from the TCA cycle. This means for every glucose we make, the TCA cycle will be turned twice, and this will generate a total of 6 NADH. This 6 NADH plus the 2 from the preparatory phase gives 8 NADH. Cashed in at the ETS, we have 8 times 2.5 to give 20 ATP. Additionally, for every turn on the TCA cycle, we have 1 FADH2 generated. This means for every glucose molecule, the TCA generates 2 FADH2. These cash in at the ETS for 1.5 ATP, and this gives us an additional 3 ATP. The TCA cycle also generates another high-energy molecule called GTP, which is converted to ATP. This means for every glucose molecule, we have an additional 2 ATP made. So let's summarize these ATP. The total we have created for processes in the mitochondrion is now 25. Let's go back and evaluate how many ATP gained in the cytoplasm are gained in the cytoplasm during glycolysis. 2 net ATP were made for one glucose molecule. So this raises the tally to 27 total ATP. What about the 2 NADH that were created during glycolysis? Well, turns out they need to be ferried into the mitochondrion to be cashed in for ATP. When this transport mechanism occurs, they lose some of their stored potential energy. As such, they can cash in for 1.5 ATP. This means these NADH will generate 3 more ATP. Now our total comes to 30 ATP. For every molecule of glucose burned in an aerobic fashion, we get on average a net gain of 30 ATP. Again, for your exam, you will need to know all 3 metabolic pathways. You need to know what you start with and what you finish with for these pathways. You also need to know where in the cell these pathways occur. Glycolysis is in the cytoplasm. The TCA and ETS are in the mitochondrial matrix. You should be able to recognize a glucose, an amino acid, a protein, a fatty acid, a nucleotide, and DNA by sight. This is not difficult to do as they all have distinct features about them. Let's start with glucose. Look at its structure at the top right of the page. It is represented in both a linear format and its traditional ring format. If you count the carbons, hydrogen atoms, and oxygen atoms, you will have a ratio of 1 to 2 to 1. In other words, there are 6 carbon atoms, 12 hydrogen atoms, and 6 oxygen atoms to give a ratio of 1 to 2 to 1. Of all the monomers and polymers I'm speaking about today, only glucose has this ratio. A monosaccharide is the monomer, and its name means one sugar. There are several types of monosaccharides, but the one we will focus on is glucose. A disaccharide means there are 2 monomers bound together through dehydration synthesis. I'll be explaining this on the next slide. Polysaccharides are the polymers of glucose. There are 3 of them that we deal with in our physiology class: cellulose, starch, and glycogen. Cellulose is the polysaccharide found in plant cell walls, and we lack the enzymes to digest it. This is the roughage in our diet. Although we gain no nutritional value from it, it does provide benefits for our body. It binds to cholesterol and helps remove cholesterol from our body when we defecate. Starch is another polymer we ingest in our diet. It is the polysaccharide found in potatoes and in some grains. This we can digest into smaller monosaccharides and absorb. Once we absorb monosaccharides, we link them together in the cells. Why? Why would it be important to take the time to polymerize many glucose molecules into glycogen? Well, turns out if we didn't, the osmotic gradient created by the cells absorbing so much glucose would cause water to enter the cell and the cell would burst. Avoiding an osmotic gradient is probably the most important reason why our cells take monomers of glucose to create polymers. Additionally, glucose is easier to store as a polymer. Here is a nice slide that shows both dehydration synthesis and hydrolysis. Let's start with dehydration synthesis. At the top of the picture you will see two monomers. In this case it is glucose and fructose. We are going to link them together to make sucrose. Notice that glucose is donating a hydroxyl group, OH, and fructose is donating a hydrogen. When the two monomers are linked, a water molecule is gained. In contrast, when we are taking a polymer, in this case sucrose, and breaking it apart into monomers, glucose and fructose, we need to use water to break it apart. This addition of water to the bond that separates the two monomers is called hydrolysis. Water is your cell's molecular scissor. For skeletal muscle and adipose tissue, glucose isn't the only metabolite that can be burned to make ATP. Lipids can also be used. Lipids have very little oxygen, the number of carbon, and, more importantly, hydrogen atoms abound. You know this to mean that the more hydrogen atoms a molecule has, the more energy it stores and the more ATP it can make. Fats generate much more ATP than glucose through a process called beta oxidation, and I will teach you this in slide 22. There are many types of lipids, and we will focus our attention on the ones written in red: fatty acids, glyceride, phospholipids, and glycolipids. The glycerides are an important fuel source for your body. It is true that carbohydrates are preferred over fats for energy, but when carbohydrates are not available, then fat is mobilized from adipocytes. First, let's talk about how fats are stored. Whenever quantities of carbohydrates enter the body that are greater than can be used immediately for energy, or stored as glycogen, the excess is rapidly converted to triglyceride, which is stored in adipose tissue. Most triglyceride synthesis occurs in the liver, but a small amount occurs in fat cells. The triglycerides formed in the liver are mainly transported by the lipoproteins to fat cells of the adipose tissue and stored until needed for energy. Carbohydrates are converted to fatty acids by their conversion to acetyl-CoA. Then the acetyl-CoA, a two-carbon chain, links together with other acetyl-CoA to form fatty acid chains that are about 14 to 18 carbons long. These fatty acids are shown here in blue boxes. Notice the abundant hydrogen atoms and realize how much fuel is stored in this molecule. The glycerol portion of the triglyceride is shown in purple, and notice it has 3 hydroxyl groups. The glycerol portion of the triglyceride is furnished by alpha glycerophosphate, which is also a product of the glycolytic breakdown of glucose. The importance of this mechanism in the formation of triglycerides is that the final combination of fatty acids with glycerol is controlled mainly by the concentration of alpha glycerophosphate, which in turn is determined by the availability of carbohydrates. When carbohydrates form large quantities of alpha glycerophosphate, the equilibrium shifts to promote formation and storage of triglycerides. When carbohydrates are not available, the process shifts in the opposite direction. The excess fatty acids become available to substitute for the lack of carbohydrate metabolism. Fat synthesis from carbohydrates is especially important because the various cells of the body have limited capacity for storing carbohydrate in the form of glycogen. The average person has about 200 times as much energy stored as fat as is stored as carbohydrate. Phospholipids and glycolipids can be used for energy, but primarily they are used to generate cell membranes. These pictures are showing you a phospholipid structure. Notice that we still see the glycerol backbone, highlighted in purple. Attached to the glycerol are two fatty acid chains. On the third hydroxyl group of the glycerol, instead of another fatty acid, a phosphate group is attached. On the phosphate group can be attached more molecules, and these change depending on the type of phospholipid created. So here they are described simply as a non-lipid group. What you should realize is that the fatty acid chains are hydrophobic and the non-lipid groups attached to the phosphate are water-loving. Therefore, phospholipids are amphiphilic or amphipathic, which means they have both water-loving and water-hating regions. A glycolipid is similar to a phospholipid except that a carbohydrate is attached to the third hydroxyl group of the glycerol backbone. These glycolipids are important as identification tags for our immune system to distinguish between self and non-self. Let me talk a little more about fatty acids. Again, note they are long chains of carbons. These chains can be anywhere from 14 carbons long to oftentimes more than 20 carbons long. And attached to the carbon atoms are many hydrogen atoms. Fatty acids are highly reduced molecules due to the plethora of hydrogen atoms and therefore store a lot of energy in these bonds. You will notice at the terminal carbon there is a carboxyl group, an acid group, and this is what allows your body to attach a fatty acid to a hydroxyl group from a glycerol. When there is a double bond between two carbon atoms, as shown at the bottom of this page, we say the fatty acid is unsaturated. If there aren't double bonds, then we say the fatty acid is saturated. The kink the double bond creates makes it difficult for many fatty acids to pack closely together, and this is the chemical reason why some fatty acids are liquid at room temperature and others are solids. Saturated fats are usually solids at room temperature, and unsaturated fats are usually liquids at room temperature. Now let's talk about essential fatty acids, ones your body cannot make but are essential to your diet. Omega-3 and omega-6 fatty acids are unsaturated, essential fatty acids that need to be included in the diet because the human metabolism cannot create them from other fatty acids. Since these fatty acids are polyunsaturated, the terms N3 polyunsaturated fatty acids and N6 PUFA are applied to omega-3 and omega-6 fatty acids, respectively. These fatty acids use the Greek alphabet, alpha, beta, gamma, etc., to identify the location of the double bonds. The alpha carbon is the carbon closest to the carboxyl group, and the omega is the last carbon of the chain because omega is the last letter of the Greek alphabet. Linoleic acid is an omega-6 fatty acid because it has a double bond 6 carbons away from the omega carbon. Linoleic acid plays an important role in lowering cholesterol levels. Alpha-linolenic acid is an omega-3 fatty acid because it has a double bond 3 carbons away from the omega carbon. By subtracting the highest double bond locant in the scientific name from the number of carbons in the fatty acid, we can obtain its classification. For arachidonic acid, we subtract 14 from 20 to obtain 6. Therefore, it is an omega-6 fatty acid. These fatty acids are essential for normal brain and eye development. In the next few slides, I will show you how we can utilize fats for fuel through a process called beta oxidation. Beta oxidation is a repeating process where two carbons are pulled from the fatty acid at a time and introduced into the Krebs cycle. Lipid catabolism involves a couple of steps. It starts with lipolysis, where triglycerides are split into the monomers that generate them, mainly glycerol and fatty acids. The glycerol can enter glycolytic pathways, and the fatty acids can enter the mitochondrion using a protein carrier called carnitine translocase. Within the mitochondrion, the fatty acids are oxidized through a process called beta oxidation. This is a repeating four-step process that ultimately creates acetyl-CoA, a two-carbon chain that can enter into the TCA cycle. Beta oxidation can generate over 100 ATP for every fatty acid. The amount of ATP generated depends on the length of the fatty acid. Unfortunately, lipid catabolism is not used as a ready source of ATP. It takes time to mobilize the lipids from adipocytes. Sadly, when you exercise, the glucose and glycogen need to be used first before the lipids are used. This is why it takes about 20 minutes of strenuous aerobic exercise before your body even starts to burn the lipids. Fatty acids are a major source of energy for most tissues. The first step in their utilization is by converting a fatty acid into a fatty acyl-CoA. This requires coenzyme A and ATP. Next comes the beta oxidation of fatty acyl-CoAs. In this process, the size of the fatty acyl-CoA is reduced in a sequence of steps. A sequence of 4 enzymatic reactions splits the molecule at the single carbon bond between the alpha and beta carbons, shown here at the end of the arrow. Long-chain acyl-CoA dehydrogenase removes two hydrogens by the reduction of FAD to FADH2, converting the single carbon bond to a double carbon bond. The FADH2 reacts with the respiratory chain, allowing oxidative phosphorylation to generate ATP. This picture is showing that 2 ATP are generated for every FADH2. I want to remind you that the ATP created depends on the energy needs of the cell. In fact, with beta oxidation, the maximum ATP would be generated. The next step is hydration. Hydrogen and OH of water is added across the double bond formed by the removal of 2 hydrogens to FADH2. The next step is dehydrogenation. Here again, 2 hydrogens are removed and added to NAD to form NADH, which reacts with the respiratory chain, allowing oxidative phosphorylation to generate ATP. This picture is again showing you the maximum ATP cash-in value of 3 ATP for NADH, and this is correct for this process. Thiolysis is the fourth step, where coenzyme A cleaves the fatty acyl-CoA between the beta and alpha carbons, splitting the 2-carbon acetyl-CoA and the fatty acyl-CoA shortened by 2 carbons. This process is repeated and shortens the fatty acyl-CoA 2 carbons at a time, and this is known as the beta oxidation spiral. The acetyl-CoA can now enter the TCA cycle, as long as there is oxaloacetate, to initiate the cycle. If carbohydrates are depleted, then the acetyl-CoA cannot enter the TCA cycle due to lack of oxaloacetate, and it will be converted into ketone bodies. Ketone bodies include acetoacetate, beta-hydroxybutyrate, and acetone. Only acetone is volatile, which means it can easily go from a liquid phase to a gaseous phase, and this gives a person sweet breath. Acetoacetate and beta-hydroxybutyrate are not volatile and end up excreted in the urine. These two are called ketoacids. So in a patient with untreated diabetes mellitus, The glucose plasma levels are high due to the inability of the insulin-dependent cells to insert glucose transporters in their cell membranes. These cells then resort to beta oxidation and amino acid use to create acetyl-CoA, which then feeds into the TCA and ultimately the ETS. However, due to depleted carbohydrates within the cell, oxaloacetate is also depleted. It is a carbohydrate derivative. This means the acetyl-CoA cannot feed into the TCA, and it is instead metabolized into ketone bodies and keto acids. This is the reason why untreated diabetes mellitus leads to metabolic acidosis. With acidosis comes also hyperkalemia, and hyperkalemia can cause arrhythmia and cardiac arrest. Again, people who don't eat enough carbohydrates or people with untreated diabetes mellitus are literally starving in the face of plenty. They lack enough oxaloacetate, a carbohydrate derivative, and cannot use the TCA cycle. Fats truly do burn in the flame of carbohydrates. When body stores of carbohydrates decrease below normal levels, moderate quantities of glucose can be formed from amino acids and the glycerol portion of fat through the process of gluconeogenesis. Each amino acid is converted to glucose through a slightly different chemical process. A low level of carbohydrate in the cells and a decrease in blood glucose are the basic stimuli that increase the rate of gluconeogenesis. Our lecture will conclude with a brief discussion on nucleic acids. Yet again, you should realize that individual monomers, called nucleotides, can be linked together through dehydration synthesis to create nucleic acids. The monomers in DNA are adenine, guanine, cytosine, and thymine. DNA is a double-stranded helical molecule shown here in the left picture. The monomers in RNA are adenine, guanine, cytosine, and uracil. RNA is a single-stranded molecule shown here at the very left. Nucleotides have a nitrogenous ring shown here in the top right picture in orange. They have a pentose sugar, that means a 5-carbon sugar, shown in the top right picture in purple, and a phosphate group shown in yellow, and the number of phosphate groups attached can vary, mono, di, or tri. RNA does undergo catabolism. RNA molecules are routinely broken down and replaced, but generally recycled as nucleic acids to make new RNA molecules. However, they can be catabolized. Let's try that again without the dog barking. However, they can be catabolized to simple sugars and nitrogenous bases, but do not contribute significantly to energy reserves. DNA is never catabolized for energy. Really, if the cell has to resort to nucleic acids to create ATP, it is already circling the death drain. This concludes our lecture on metabolism. For testing purposes, you should know the metabolic pathways described in this lecture, be able to recognize monomers and polymers by sight and description. Additionally, you should understand dehydration synthesis and hydrolysis.